Hello friends, I am Dr. Prashant Sharma and you are watching Medicos Hub. In this video we are going to discuss about the antigens. First of all, what is antigen? Anything that stimulates antibody generation can be termed as antigen. So antigen is antibody generating substance. These antigens may be whole organism like bacteria, virus, etc. or these may be part of organism like cell wall, capsule, flagella, or these may be non-microbial substance like pollen, egg white, mismatched blood cells, or transplanted tissues. Now the chemical nature. Chemical nature is usually protein, but these may be nucleoproteins, glycoproteins, and polysaccharides. Properties of antigen. There are two important properties of antigens. First one is immunogenicity and another is antigenicity. Now immunogenicity means it is the ability of antigen to stimulate the cellular or humoral immunity, while the antigenicity is the ability of an antigen to interact or react with the products of cellular and humoral immunity. Now types of antigen. These are also of two types. Complete antigens. Complete antigens means immunogenicity present, antigenicity present. So those antigens which possess both these properties are known as complete antigens, while the partial antigens, which are also termed as haptens, lack immunogenicity. Now, biological class of antigens. There are two major classes of antigens. First one is T-dependent antigens. These are also termed as T-D antigens. Another is T-independent antigens, also known as TI type of antigens. Now we will differentiate these two major categories of antigens. The chemical nature. These antigens are structurally complex proteins. These are structurally simple. These are lipopolysaccharides. These may be flagellar components or cell wall materials. Now immunologic or immunogenic response. Here is wide range of doses. It is over wide range of doses, while the immunogenic response for T independent antigens is dose dependent. Then memory is present, while here the memory is absent. Antigen processing step is required in case of T-dependent antigens, not required for T-independent antigens. If metabolism is considered, the TD antigens are rapidly metabolized, while the TI antigens are slowly metabolized. These activate B cells. Both of these activate B cells, but important thing is that it activates mature B cells, and the activation is monoclonally, while the T-independent antigens activate both immature and mature B cells and polyclonally by manner. The stimulated B cells undergo class switchover and affinity maturation. Yes, but it doesn't occur in TI type of antigens. Antibody produced are of all classes, while this is restricted to IgM and IgG3 type. So these are the two major categories of antigens. Another important category is super antigens. Now what are these super antigens? Super antigens can be considered as third biological class of antigens. These react or interact with APCs and T cells. Here, endocytic processing is not required. We have discussed about that. In case of typical antigens, endocytic processing is required, but in case of super antigens, it is not required. Now, what is the mechanism of action? The mechanism is super antigens bind with MHC class 2 molecules of APC and V beta domain of T cell receptor. Because of this activation of large number of T cells occur, and it's important that this number is very high in comparison to the common antigens. Next is massive cytokine release and immune modulation. So this is the mechanism of action. Now examples of such super antigens are Staphylococcus, which secrete enterotoxins, then HIV, rabies virus. These all are examples of super antigens. Now there is a term that is epitope. What is that? It's very important term, epitope. Epitope is antigenic determinant site of antigen that binds with the paratope of antibody to initiate the immunological or immunogenic response. It is the smallest unit of antigenicity. Its molecular weight is between 600 to 1000 Daltons and its size is in range of 25 to 34 angstrom. So this is epitope. Another important term is adjuvant. Adjuvant is itself non-antigenic, but when it combines with a particular antigen, the antigenicity of that antigen is increased. Antigenicity. So this is adjuvant. So this is all about the antigens. Hit the like button, share and subscribe our channel to get the latest updates and notifications.